Covalent modification is one of the method for enzyme regulation. You know that the enzymes are proteins. These are nothing but proteins which acts as biological catalyst. Biological catalysts are those which catalyze the reactions in the cells, and these are nothing but enzymes. And you also know that all enzymes are proteins, but all proteins are not enzymes. Proteins can also function in different aspects, like some proteins act as blood proteins, some act as giving structure, some proteins act in transporting some of the molecules. Okay, with that introduction, we will go to covalent modification. These covalent modification refers to the forming and breaking of the covalent bonds. You know what are covalent bonds in chemistry, which are formed by the sharing of electrons, and these covalent attachment of another molecule can modify the activity of enzymes and many proteins. Due to the covalent modification, that means either forming or breaking of covalent bonds, what is happening here? They can modify the activity of enzymes. That means they can make the enzymes or proteins to be active, or they can also make the enzymes to be inactive. They can do both the things by simple covalent modification. The donor molecule provides a functional group that modifies the properties of the enzyme. These modifications usually they are reversible in nature. If we see the examples of covalent modification, the most common example or the aspect what we can see in the covalent modification is phosphorylation and dephosphorylation, which are the most common covalent modifications. Along with the phosphorylation and dephosphorylation, it is not the only means, but there are some other modifications. What are they? Acetylation and deacetylation of histones is also one of the example for covalent modification. You know that the histones they wrap the double-stranded DNA. If this is the DNA, these are the histones which are wrapping the DNA. So these histone proteins they can be either acetylated, that means acetyl groups are added, CH3CO groups are acetyl groups, they can be added to the histones, or they can be removed. These acetylated groups they can be removed, and this is also one of the example for covalent modification. This covalent modification, that means acetylation and deacetylation, they assist in the packaging of the DNA. That we know that the DNA, it is double stranded DNA, but it do not remain all the time in the double stranded nature. It is packaged on the histone proteins, later higher order of packing into the chromosomes. And for this packing, the histone proteins play a major role in the packing. And these histone proteins they are either acetylated or deacetylated. What is the importance means? Highly acetylated histones they are associated with the genes which are actively transcribing. That means in the DNA also we will see some of the genes are very actively transcribing. That means from which we get the RNA is nothing but transcription. So there are some genes from which the RNA is actively transcribing. In those genes, what type of histones are present? Acetylated histones are present. Highly acetylated histones, they are examples. They are present in the actively transcribing genes. Acetyl transferase and deacetylase enzymes are regulated by the phosphorylation. So in order to acetylate these histones, what type of enzymes are required? Acetyl transferases, which these are the enzymes which transfer the acetyl groups are nothing but acetyl transferases. These acetyl transferases are required to acetylate the histone proteins. Likewise, deacetylase enzymes are required to remove the acetyl groups from the histones. They are also regulated by the phosphorylation. That means in the phosphorylation and dephosphorylation only, this acetyl transferases and deacetyl enzymes, they are also examples. Let's see the examples, actual examples, enzymes which function by covalent modification. Or the best example is glycogen phosphorylase enzyme, which is also due to the acetylation of histones. And another example is ubiquitination of ubiquitin cyclin, which controls the cell cycle. So ubiquitin is attached to some of the proteins, thereby they are undergoing the cell death. For that also the phosphorylation process is very very important. That means covalent modification is important. The covalent modification of proteins is a potent means of controlling the activity of enzymes and other proteins. So they control, that means it is the method for enzyme regulation. Phosphorylation is the most common type of reversible covalent modification, which I already told you. The signals can be highly amplified by the phosphorylation because a single kinase can act on many target molecules. Why phosphorylation is a very important means or very common means if a single protein is phosphorylated, for example, if this single protein which is phosphorylated, this can target many proteins. This can target many proteins, thereby a single enzyme can amplify the its effect on different target molecule. These are all the target, 5 to 6 targets or more than many targets are present, which can be uh, functioning, whose function depends on the phosphorylation of one particular enzyme. That is why it is the most potent means of controlling the activity of enzymes. Certain enzymes exist in the active form as well as inactive forms, which are interconvertible depending on the needs of the body. Okay, so depending on the need of the body, our body knows that when the enzyme has to be synthesized, under which circumstances the enzyme has to be active. That means it has to perform its function. When it is not needed, the enzyme should be in the inactive condition, inactive form. So there should be switching between the active form and inactive form. There should be switching between the inactive form and the active form. This is interconversion. This interconversion is brought about by the reversible covalent modification. And this reversible covalent modification is mainly by means of the phosphorylation and dephosphorylation, or either by oxidation and reduction of the disulfide bonds. Disulfide bonds means here a bond is present like this between the two cysteine molecules of different polypeptide chain. This is nothing but disulfide bonds. So the oxidation, it can undergo the process of reduction where the disulfide bond is broken down and two polypeptides, they can be separated. Okay, so phosphorylation and dephosphorylation as well as the oxidation and reduction of the disulfide bonds, they are also examples for the reversible covalent modification. Thereby the enzymes can be either active form or inactive form. Let's see the main enzyme example, which is nothing but glycogen phosphorylase. This is the muscle enzyme which breaks the glycogen to provide energy. This glycogen, what is glycogen? Glycogen is the storage food material in case of animals. Many glucose molecules, they are joined to form the glycogen. Here, this particular enzyme, which enzyme? Glycogen phosphorylase enzyme, it breaks the glycogen. When the glycogen is broken down, the molecule which is formed is nothing but glucose. The molecules which are formed is nothing but glucose. This glycogen phosphorylase, it has to operate only under certain conditions. When it has to operate, when there is no energy in the cell. This glycogen phosphorylase, it is a homodimer. Homodimer means what? It contains two subunits. The enzyme contains two subunits. That too, they are homo, means identical. Two identical subunits, if they are present, it is called as homodimer. Glycogen phosphorylase enzyme is a homodimer, and this exists in two interconvertible forms. There are two interconvertible forms. One is the active form, another one is inactive form. The active form is known as phosphorylase A form, and that means glycogen phosphorylase A form, and the inactive form is glycogen phosphorylase B form. What is inactive form? It is the glycogen phosphorylase B form. What is the active form? Glycogen phosphorylase A form. Glycogen phosphorylase B is D-phosphoenzymes. That means it do not contain phosphate. Phosphate is not attached to the phosphorylase B. That is why it is inactive. Whereas the active form is phosphorylated form. Phosphate is attached to the glycogen phosphorylase A. That is why it is in the active form. Active form means what? The active form of the enzyme. For example, glycogen phosphorylase A form, if it is active, this A form could catalyze this reaction. What is the function of glycogen phosphorylase? We already know that the glycogen phosphorylase is the enzyme which breaks the glycogen to provide energy. So this enzyme, it breaks the glycogen into glucose molecules. For that one, the glycogen phosphorylase A is functional but not B. Why the B form is existing? Glycogen need not be always broken down. That means we always do not require the breakdown of glycogen. When the glycogen should be broken down, when we don't take food or when we are in starving conditions, then only the glycogen has to be broken down. Then only the glycogen phosphorylase enzyme should be in the active form. Otherwise, if we are taking sufficient amount of food, there is no need for the breakdown of the glycogen, and the glycogen phosphorylase enzyme, it should be in the inactive form. See here, the glycogen phosphorylase B, which is the D-phosphoenzyme, is inactive. This inactive form is converted to active form of glycogen phosphorylase A by what process? By the phosphorylation of serine residue. Where this serine is present, phosphorylase, glycogen phosphorylase is the enzyme. It is the enzyme. It contains many amino acids. Likewise, it also contains serine amino acid. Here, the phosphate group. The phosphate group, it is attached to the serine amino acid. That's the meaning of this sentence. Here, the phosphorylation of serine residues. Serine is part of the glycogen phosphorylase enzyme, to which actually the phosphate group is attached. The inactive form, that means glycogen phosphorylase B, is produced on dephosphorylation. Suppose if phosphate group is attached to this enzyme, that means this is the active form. Okay, this phosphate group, if it is removed, for example, if the phosphate group is removed, this phosphate group is removed here, then what happens? This glycogen phosphorylase enzyme, now it is the inactive form of the glycogen phosphorylase. Then it cannot catalyze the formation of glucose from the glycogen. These are reversible or interconvertible forms of glycogen phosphorylase A as well as glycogen phosphorylase B. This figure, it very clearly illustrates what I said. The glycogen phosphorylase enzyme exists in the two forms, glycogen phosphorylase A, which is the active form, and glycogen phosphorylase B, which is the inactive form. The inactive form is D-phosphoenzyme. D-phosphoenzyme means it do not contain phosphate groups, whereas the active form it contain phosphate group. It contain phosphate groups are present. That is why it is the phosphoenzyme. This glycogen phosphorylase B, glycogen phosphorylase usually exists in the inactive form, which is D-phospho form. This glycogen phosphorylase is now converted into the active form. When it is converted, when it is necessary, when the glycogen has to be broken down, it should be converted into glycogen phosphorylase A. How it is converted? It is converted by an enzyme called kinase, a protein kinase. Kinases, remember, these are the enzymes which particularly takes up the phosphate from the ATP. That means they break down ATP, take the phosphate group and transfer the phosphate group to the substrate. What is the substrate in this case? This enzyme itself is the substrate. Which enzyme? Glycogen phosphorylase B is the substrate. And what is the product? Glycogen phosphorylase A is the product of which enzyme? Kinase enzyme. Kinase, a protein kinase enzyme, what it does is it breaks the ATP, takes up phosphate, and it transfers the phosphate from ATP to the glycogen phosphorylase B so that it is converted into glycogen phosphorylase A. These phosphates, look here, these phosphates have transferred to the inactive form so that it is converted into active form with changed conformation. This active form, now it can catalyze the breakdown of glycogen to glucose. But when this process is not needed, sufficient amount of glucose is present in the cytoplasm, then what happens? The glycogen phosphorylase enzyme, it is not needed. Phosphorylase A enzyme, that means the active form is not needed. Then immediately what happens? This active form should be converted into inactive form. How it is converted into inactive form? Just by removing the phosphate groups. These phosphate groups, when they are removed, here, see here, these phosphates group are removed. These phosphates group are removed by the enzyme, phosphatase enzyme. Then the active form, glycogen phosphorylase A form, it is converted into inactive form of glycogen phosphorylase B. Hope it is clear to you. The other examples here, in this particular case, when the enzyme is active in the phosphorylated form or dephosphorylated form. Remember, in the phosphorylated form. In the phosphorylated form, the enzyme is active. Okay. The other enzyme, other examples of phosphorylated active enzyme. In the previous case, what is the active form? Phosphorylated. Okay, phosphorylated active form. Here there are also some other enzymes which are phosphorylated active enzymes. What are the other examples? Citrate lyase, fructose 2,6 bisphosphate. Both are active. When they are active, in the phosphorylated form. In the phosphorylated form, these enzymes are active. That is why they are called as phosphorylated active enzymes. What are the examples? Citrate lyase and fructose 2,6 bisphosphatase are the examples. There are some enzymes which are active in the dephosphorylated form. There are some enzymes which are active in the dephosphorylated form and become inactive when phosphorylated. Examples are glycogen synthase, acetyl-CoA carboxylase, HMG-CoA reductase. These are examples of the enzymes which are active when they are active in the dephosphorylated form. When the phosphate is removed from those enzymes, then they are active. Okay, these three enzymes, glycogen synthase, which is reverse to that of glycogen phosphorylase, acetyl-CoA carboxylase, HMG-CoA reductase. These are examples for the enzymes which are active in the dephosphorylated form. And these are the enzymes which are active in the phosphorylated form. What are they? Citrate lyase, fructose 2,6-bisphosphatase, and glycogen phosphorylase. These are the enzymes which are active in the phosphorylated form. Hope you understood the discrimination between the dephosphorylated active and phosphorylated active. Yes, now it is very clear to you. We have seen what is meant by covalent modification and the enzymes which are regulated by covalent modification.